Namaskar, dear viewers. Welcome to today's session of critical care webinars. And I have a very special guest today, Dr. Sumit Ray, sir. Sir is a very eminent figure in the Indian critical care setup. And today he is going to be speaking on precision medicine in critical care, possibility or pipe dream. So you have all been hearing about the impetus on precision medicine and that protocols and guidelines are not really something which are sacrosanct. So he'll be speaking on that. And to introduce Dr. Sumit Resa to you, he is head of the critical care department and medical director of Holy Family Hospital, New Delhi. He has been the ex-vice chancellor and ex-secretary of the Indian College of Critical Care Medicine. He was a former secretary for examinations for the Indian College of Critical Care Medicine, and he's a well-recognized examiner and teacher for critical care medicine. And he was awarded the Chikitsa Ratna Award by the Indian Medical Association, Delhi, for contribution to medicine and humanity. So, sir is very intricately associated with critical care, education, and examinations in our country. So, with that, sir, I will hand it over to you to take us through the next 45 minutes or so.
Thank you, Tapesh. I hope I'm audible, and thank you everyone for being there on a Sunday afternoon. Good afternoon, everyone. So, precision medicine, is a possibility, or is it just a pipe dream for now in critical care medicine? So, okay, so slides, yeah. So, precision medicine, the definition of precision medicine by the US FDA and the NIH states that it is an innovative approach to tailoring disease prevention and treatment that takes into account differences in people's genes, environments, and lifestyles. The goal of precision medicine is to target the right treatments to the right patient at the right time. And that is probably everyone's goal. But it is, this definition brings to light the focus that is probably going to shift in the not so distant future on how we treat patients rather than large groups of patients being treated in the same manner, and that one size does not fit all, and there may be certain subsets of or subgroups of or endotypes of patients who may need different treatments. That is where precision medicine is probably heading towards. The early advances in precision medicine have largely occurred in oncology, from where this has come, where diagnosis and treatment are increasingly based on genomics. The examples, the positive examples from oncology are the HER2 positive breast cancer treatment, the EGFR positive lung cancer, which highlight the potential of precision medicine. So in critical care, through this talk, I would be looking at the possibilities, the challenges that are there, and the concerns that precision medicine raises, and focusing primarily on two major disease processes and two killers in critical care, sepsis and ARDS. Those are the two main focus areas I would be touching upon. So there are certain terms that we need to know about before we go on to precision medicine. Is the phenotype, which probably all of you know is basically a clinically observable set of clinical features. Like syndromes such as ARDS and sepsis would be considered as phenotypes. Subgroups are any subset of patients within a phenotype which could be defined using certain cutoffs or clinical variables or measures, like the PF ratio for the for the ARDS definition, for the Berlin definition and global definition. Subphenotypes are distinct subgroups of a phenotype based on shared sets. Shared sets are patterns of observable or measurable properties which can be reliably discriminated from other subphenotypes. An endotype is a form of subphenotype with distinct functional or pathological mechanisms that may or may not be associated with a specific treatment response. Which is an endotype can also be like a treatable trait. That means a set of clinical characteristics or biomarkers indicative of a pathogenetic mechanism that responds to specific interventions within a clinical syndrome. Like, for example, a hyperinflammatory or a hypoinflammatory or a immune paresis in ARDS and sepsis. They would probably, the subsets would have certain treatable traits which can be focused upon. Another important concept is the heterogeneity of treatment effects and enrichment. What is heterogeneity of treatment effects? It basically tells us about the difference in the treatment response in a group due to a variability in response of a phenotype to drug response or treatment response. And enrichment is a continuous kind of a correlation to that is that once you have a subgroup or a subphenotype which is which responds to a treatment and another subgroup subphenotype which does not respond to a treatment, when you are doing a study, you can do an enrichment study, which means that strategizing to reduce the number of heterogeneity in the sample size and making more homogeneous populations with the similar risk profiles being targeted for the research with the specific intervention. So these two are important elements, the heterogeneity of treatment effects and enrichment, important parts of precision medicine. So if we look at precision medicine, the possibilities that we have to focus on are we have to look at the genomics and epigenomics. That I'll come to that in a little while. Transcriptomics, metabolomics, and proteomics. These probably will help us to differentiate sepsis from SIRS, prognosticate the sepsis, the severity of illness, the susceptibility to sepsis, the response to treatment, and also bioinformatics, which is not part of the omics, but basically using large data sets to predict clinical trajectories, to predict the response to treatment. So I'll go into all of these a little bit. So if you look at genomics, all of you know is the study of genes and their functions. Basically, if you look at sepsis or ARDS, the various polymorphisms that have been studied, which have been described in genes that encode, like in sepsis, a pro-inflammatory versus an anti-inflammatory cytokines, cytokine receptors, cellular recognition pathways, intracellular signaling pathways, and hemostasis of molecules. Epigenomics is the study, the additional changes that alter gene expression without changing the DNA sequence. And here they looked at DNA methylation, the non-coding RNAs, etc. And the epigenetic modifications can respond to environmental stimuli. And epigenetic biomarkers could help detect patients prone to deterioration and unfavorable evolution. So there were certain studies by Wong et al., and he has focused on pediatric sepsis, and they looked at the genome-wide expression of profiling using whole blood-derived RNA from about 100 children in septic shock children with septic shock, and they, based on this genomic expression, they were able to differentiate into three subclasses. The subclass A had a repression of genes corresponding to adaptive immunity and glucocorticoid receptor signaling, just an example. And these patients did worse than the subclasses B and C. I'm not going to detail of the subclasses B and C, just tells us that there could be possibility by using genome-wide expression profiling to look at which is the, based on the genomic profile, how is going to be the response of patients to different forms of septic shock and their response to therapy. Transcriptomics is the transcriptome is a set of mRNAs and non-coding RNA molecules in a specific cell or tissue. The transcriptomics basically aims at monitoring gene activity and regulation. And Sweeney et al. Did a transcriptomic profile of 700 septic patients and found that there were three subtypes based on transcriptomics. The adaptive subtype, which was associated with a lower clinical severity and lower mortality, the coagulopathic subtype, and the inflammopathic subtype, which had more frequent septic shock. Metabolomes, that is what is more easily studiable at this present moment, and we actually are doing a study on this. The metabolomes are a collection of small molecules produced by cells, and critical illness alters this metabolomic profile in a patient of in severe illness like sepsis, ARDS. And these metabolomes are used to identify biomarkers, drug activities, drug-induced toxicity, and metabolism that happens. And these metabolic studies in sepsis can be used to discriminate patients with sepsis versus non-infectious SIRS with an inflammatory response, prognosticate the disease process, recognize are these patients responding to treatment or not. Then there is proteomics. Proteomics is the study of the proteome, which consists of all the proteins expressed by the genome of a cell, tissue, or organism at a given time under certain conditions and environment, like a certain conditions of stress that you see in a patient of sepsis or ARDS. Proteomics will probably help increase our understanding of the pathophysiology of disease, identify new molecules that can predict patients' evolution and prognostic factors. Then we come to the non-omics, which is the bioinformatics. Basically, in omics-based studies are still too slow to be useful in ICU for clinical decision making, rapid ICU clinical decision making. So we need to look at large sets of data. Basically, for the near future, along with the omics, what is going to be important is looking through large data sets, which is derived from not just the omics platforms, but also from randomized trials, also from large observational trials, physiological variables from electronic medical records, which can give us insights into the pathophysiology of the critical illness that we are looking at, sepsis or ARDS, identifying distinct subphenotypes or endotypes within those, and also furnish an infrastructure upon which to generate and test new hypotheses, do new research based on that. And these huge data sets are, those are the possibilities and also those are the challenges. And in these data that we need to analyze requires a lot of, both integration and cooperation between organizations, and open source electronic data capture tools will be invaluable. In this, starts have been made, like one of the classic is the Mimics-3, which is the Mimics-3 data is basically a data descriptor, which it's a database of the Beth Israel Deaconess Medical Center in Boston, and it is open source. Basically, it integrates de-identified comprehensive clinical data of patients who are admitted in this hospital. It's open in nature and could be useful internationally. Anybody can access it for academic purpose, for industrial research, for quality improvement initiatives, and higher education coursework. So what it does is that there is data, which is the ICU. Basically, it's focusing on the ICU. So it has the bedside monitoring data, vital signs, waveforms, trends, alarms, the orders, the fluids, medications, progress notes, the hemodynamics, the demographics of patients, the radiology, the imaging, etc. All of them integrated in this data, and even the billing and the procedures done. Okay, all that is put in a data archive, and they're de-identified. There is no patient name associated with it, and patient identification data is taken away, and this is put in the Mimics database, which can be utilized by other organizations, other researchers for their own research, and also from that data develop their own models to do research in their own centers, and also give feedback to how to improve this data integration and collection. And what it ends up doing is that these kind of knowledge networks, which would need data from clinical data, as we said, EMR data, imaging data, all the omics data, right? Along with data from randomized controlled trials, observational studies. These data would help us identify the phenotypes or subphenotypes, the inference studies, the causal inference studies, explore biomarkers, detect the heterogeneity of treatment effects, subgroups which respond differently. And from these, identify candidate subgroups, which can be further studies, which could potentially respond or not respond to the treatment that is designed. And further take it from these candidate subgroups, use what is called adaptive enrichment strategies, and I'll come to that, to try to design studies where less heterogeneous subgroups are studied to look at the benefit or otherwise of the interventions that we are planning to devise. So there is this very good article in AJR CCM in October 2021, which is the research agenda, which is the article is called the Research Agenda for Precision Medicine in Sepsis and ARDS. And this is the official American Thoracic Society research statement. The first recommendation they gave is to create large, richly phenotyped, harmonized knowledge networks of clinical imaging and multi-analyte molecular data from patients with sepsis and ARDS. For that, there are already, as I said, the Mimics data is there. Then there is the NHLBI, that's the ARDS network data, where clinical data, biomarkers are measured within the RCT testing systems in ARDS. Then there is in sepsis, there is the phenomes data in pediatric critical care research network. There is the IDBJ, which is the integrated central biobank for sepsis from 3 previous sepsis biobanks. And for COVID, you have already seen what has been the kind of data networks and the knowledge networks that have been created. Along with that, the second recommendation is to implement novel trial designs to identify precision medicine strategies for sepsis and ARDS. Some are the adaptive trial designs that I spoke about, or the adaptive enrichment, which basically means that constant modification of the eligibility criteria to target participants to likely benefit and or exclude participants unlikely to benefit from the intervention that we are doing. By analyzing these data sets, look at a more homogeneous sets of patients to focus on for that treatment subgroup. And the same could be done with biomarker guided or biomarker threshold adaptive design, which is the same thing with biomarker guidance. Or you could have the platform trial, where multiple interventions are tested within a single trial framework against a single control arm for a single condition, and the therapeutic arms are added or dropped from the trial based on interim analysis to see if it's working or not. We saw that in COVID very well. The recovery, the REMAP-CAP, the Solidarity, the iSpy COVID were all platform trials. And recovery and REMAP-CAP was very successful models, and we'll come to that. Then the other recommendation is advanced data science and engineering approaches to facilitate precision medicine strategies. So that means basically machine learning, data collection, data analysis science has to be developed, which could be utilized from the engineering models to data science models into medicine. And also to develop tools for real-time application of these to analyze the data and quickly utilize it to your specific patient subgroup, which you are treating right now. So if you look at the platform trials, the recovery trial, what did it do? Basically, a platform trial is that you have, basically one arm, which is, basically you have a single there's a single control arm, right? And against that, you are trying out different treatments. You have the usual care alone on one arm, and the other interventions are being added and removed depending on what subgroup analysis has shown. Is the patient responding or not, patients responding or not? And this is what happened during COVID, and we found many therapies which were useful and many therapies which were discarded in the process of treating COVID. So along with that, so till now, most of the time, the bench to bedside kind of research has targeted that you have animal studies or lab studies, which have been extrapolated to more translational studies to patients, to clinical trials, and the data collected from the RCTs. Either there is a successful RCT trial, which, that treatment becomes useful and it is utilized, or depending on the subgroup analysis, there is, we find that there is a subphenotype which can be treated better or that responds better to the therapy, while other subphenotypes do not respond very well. Then more studies are planned focusing on this responsive subphenotype. So this is called forward translation. Now they are focusing on reverse translation. That means you look at the response versus the non-response heterogeneity, the heterogeneous groups which respond differently, the molecular mechanisms involved in the drug effect or the treatment effect, and then again go back to the bench, go back to the lab studies to identify which would be a better target. So this is called reverse translation. So not only use forward translation, but reverse translation, which would be useful in precision medicine. So one of the things that we are doing in our center, which we have applied for an ICMR grant, 2 studies actually, one in ARDS and one in sepsis. The ARDS study I'll come to later is already there are identified subphenotypes which are there, which respond differently to the hyperinflammatory and the non-hyperinflammatory subphenotypes in ARDS. So they have been identified by latent class analysis, and I'll come to that. And we are looking at those to do a prospective trial on that. That's how forward translation works, and I'll come to that in a little while. So let us look at some of the successes that have been done to that have been possible in identifying the clinical subphenotypes. For example, this study by Seymour et al. For sepsis. So they looked at a derivation data of almost 20,000 patients from 12 centers in Pennsylvania hospitals. And the validation cohort was about 43,000 patients. So from the emergency, they basically did a sepsis endotyping in emergency care, the Seneca trial, and looked also at the genetic and inflammatory markers of sepsis in another trial and correlated it to the derivation cohort in the validation cohort, and found, using a huge data analysis, huge amounts of data of these large volumes of patients, clinical variables, lab variables, physiological variables, all together, huge data sets to analyze, and they found 4 phenotypes. The alpha phenotype in sepsis, where the mortality was lowest with the lowest administration of vasopressors. The beta phenotype, which had patients who were older, had more chronic illness and more renal dysfunction. The gamma phenotypes, which had patients with more inflammation and pulmonary dysfunction. The delta phenotype, which had patients with more liver dysfunction and septic shock. And these patients, these 4 phenotypes behave differently. The patients, the number of days on ventilator, etc., was higher in the gamma phenotype, which had more pulmonary dysfunction, obviously. Administration of vasopressors were higher in the lowest in the alpha phenotype. And in-hospital mortality, if you see, was the highest in the phenotype which had liver dysfunction with septic shock, compared to the least, which was in the alpha phenotype, and in the medium range was the gamma phenotype. So they did, were able to describe with these chord diagrams the different subphenotypes which would respond differently. They had different outcomes. But what is more interesting is they looked at all these phenotypes and assessed certain pre-existing trials against these phenotypes. So they looked at, suppose, this example of the process trial, which is an EGDT trial, right, of almost 900 patients. And the phenotype distribution in their analysis was about, this is the percentage, alpha, beta, gamma, and delta, if you can see on the left side. And the overall response the possibility was harm. But then they did a simulation that if there was 0 alpha phenotype, that alpha phenotype was the one with the least vasopressor requirement. If there was 0% alpha phenotype and the other phenotypes were dominant, then the possibility of harm was very high. And if the simulation, as there was an increasing percentage of alpha phenotype, if the alpha phenotype became 100%, that in alpha phenotype subphenotype patients, the possibility of benefit was higher for the early goal-directed therapy for the process trial. And they looked at the delta phenotype. So again, if it was the usual distribution as they found of the phenotypes, then the possibility of harm or benefit was not sure. But if the delta phenotype increased, the percentage of delta phenotype increased to 25%, the possibility of harm increased. And if it was close to 50%, the possibility, if there were more delta phenotypes, then the possibility of harm with early goal-directed therapy with higher volumes of fluid would be higher. Okay, so the subphenotype that we could focus on, if we knew which subphenotype this patient belonged to, then our treatment would vary, how much fluid to give or not give. So this was another analysis of the VANISH trial, the transcriptomic signatures. And they did a post hoc analysis of the VANISH trial, which was the trial of vasopressin and hydrocortisone in septic shock patients. And looking at the 7 discriminant genes that they had, they had determined in a previous trial, the 7 genes, they found 2 subphenotypes, the SRS1 and the SRS2. The SRS1 was the immunosuppressed endotype, and the SRS2 was the immunocompetent endotype. And they found that in the VANISH trial, that there was no difference in the 2 SRS groups, the immunocompetent and the immunosuppressed, for assignment to vasopressors and the outcomes. But what they found was that hydrocortisone, in the immunocompetent endotype, giving steroids, hydrocortisone, was associated with higher mortality. That means if the patient was immunocompetent, was able to produce probably more, the HPA axis was okay, there was no immune paresis and immune suppression, then probably steroids were not a good idea. But in patients who were immunosuppressed, they probably would benefit better with low dose hydrocortisone. So the subphenotype that you wanted to treat would respond differently to steroids in sepsis. This was another trial looking at, again, genomics to look at different pathways and mechanisms in sepsis, and they identified 5 different subsets which responded differently to different therapies. But in sepsis, there are different forms of response. One is the 4 subphenotypes that we looked at, the alpha, beta, gamma, and delta. But there could be a pro-inflammatory phenotype versus an immune paresis phenotype or a mixed response phenotype. And more evidence suggests that initially, most of the patients are now who have a hyperinflammatory response usually survive with resuscitation. If they die early with unbridled hyperimmune response, not a usual phenomena anymore. But a large percentage of patients have a immune dysregulation of innate immunity. They are in persistent in low-grade inflammation, who there is poor tissue regeneration, long-term death is higher, they have recurrent infections. You would see patients having repeated bouts of nosocomial infections. And then there is the adaptive immune suppression subgroup, again, very prone to repeated opportunistic infections, long-term deaths, particularly in the vast majority of elderly patients, where with multiple comorbidities, who we may be able to help them survive short-term, but long-term, in terms of several weeks to years, they usually die from the initial trigger of a sepsis. So there are different patterns of immune response. We had actually studied this and published in 2014 in Critical Care. And this we were looking at the pro versus the anti-inflammatory cytokines. The pro-inflammatory cytokines, we looked at 3 cytokines, TNF alpha, interleukin 6, and interleukin 8. And the anti-inflammatory was interleukin 10, interleukin receptor antagonist. And we found that the ratio between the pro and anti-inflammatory cytokines gave us an idea about mortality and outcomes. What we found was the mortality was significantly higher in the anti-inflammatory subgroup, which had a predominant anti-inflammatory response early on day 1 and day 3. And the mortality was 49% almost versus 23% in the initial pro-inflammatory group. And also the median day of death was significantly delayed in patients of anti-inflammatory who got more secondary infections, more nosocomial infections as compared to the pro-inflammatory group. The nosocomial infections were not because, yes, it could be because of their infection control practices, but it was also, they were set up, the patients were set up for nosocomial infections because of their immune response. And this is what Hotchkiss et al. Had studied earlier. And this is also expressed in different ways by the low monocyte HLA, human leukocyte antigen DR expression, the monocyte HLA DR expression. That has also been studied, and we are studying that. This is part of our trial that we have planned. We have applied for an ICMR grant. What these people looked at, what we are also going to look at is not only the cytokines, but the expression of the monocyte HLA DR, which gives us an expression. If there was lower levels of monocyte HLA DR, those are the immuno-incompetent individuals who tended to have higher mortality and more nosocomial infections. So the whole idea is in precision medicine would be that if you have the heterogeneous subgroups, low-risk patients like patients who have are immunocompetent, who have low vasopressor usage, right? Low could be treated by usual care like high fluid resuscitation, vasopressors, antibiotics. While those with high-risk patients who are not immunocompetent or have more liver dysfunction, have more septic shock, probably in those patients, early vasopressors, lower amounts of fluid resuscitation could be useful. So you could tease out the subgroups of how to treat different subgroups. That would be the focus of precision medicine. Looking at ARDS, all of you may have seen this study by Caroline Calfee et al., which was the latent class analysis of 2 RCTs, the ARMA trial and the alveolar trial. And from there, they determined the 2 phenotypes, the subphenotypes, the phenotype 2, which was the hyperinflammatory phenotype, which had higher incidence of shock, acidosis, higher concentration of inflammatory biomarkers. These are the ones. While the phenotype 1 was the hypoinflammatory, or the not hypoinflammatory, but the non-hyperinflammatory subphenotype. And they found that when they analyzed the data, the hyperinflammatory phenotype had significantly higher mortality in both the cohorts. You see, significantly higher mortality, less ventilator and organ failure free days. Okay. But what is more important for me, and I keep saying this, is that how they behave to other therapies. Phenotype 1, the patients with less amount of shock, and phenotype 2, the patients with more amount of shock and acidosis. They behave differently to P. So in the phenotype 1, less inflammatory phenotype did better with low PEEP. If you can see, the mortality was 16% versus 24% in the high PEEP group. They had better mortality, less mortality rather, and more ventilator and organ failure days. While the phenotype 2 with more hyperinflammatory, with more shock, actually did better with high PEEP. That means they had a more leaky endothelium. They had, both vascular and alveolar endothelium. So they did better with higher PEEP. So that's why just don't go by the fact that the patient is in shock and don't reduce your PEEP. Titrate the PEEP in this subset of patients. Okay. More analysis, latent class analysis now of ARDS with the FACT trial, about 1,000 patients. So the same group defined the phenotype 2 with even less number of biomarkers. They found that even 3 biomarkers could be enough along, right? And the phenotype 2, the same phenotype, the same higher mortality. But what they found in the analysis of the FACT trial was that the subphenotype 1, that means the less inflammatory phenotype, did better with liberal fluids as compared to conservative fluids in the FACT trial. They did less mortality. While the phenotype 2, which had more shock, did better with less fluids so they did better with conservative fluid therapy as compared to a liberal fluid therapy, the subphenotype 2. So already we are able to tease out, if we are able to, and this is another study which we are doing, an ICMR project which we have applied for, looking at almost 30 centers, where we'll be looking at these 2 different phenotypes, taking back from what has been derived by Carolyn Calfee's group, the inflammatory phenotypes, and from there define how we treat in a forward manner, how we treat these patients. So if we look at precision medicine for ARDS can be phenotyped based on 3 domains. Etiology, what is the cause, the initiation of the lung injury, the amplifiers of lung injury. And the biology, that means the host response, the endothelial injury, the angiopathy, or the physiology. Okay, how is the respiratory drive? How severe is the hypoxemia? How is the respiratory mechanics? And based on that, different subgroups could respond differently to steroids, to antimicrobial agents, to antifibrotics, or to PEEP strategy, recruitment maneuvers, prone positioning, sedation and analgesia, immunomodulation, endothelial protection, again, fluid management, etc. So these are the phenotypes we can tease out from among ARDS subgroups. Studies have already started in AKI also, acute kidney injury in critically ill by latent class analysis, and I'm not going to go into that. So those were the possibilities. Which are the areas we can possibly focus upon? But what are the challenges? The challenges are finding discrete groups and subgroup of patients. And there are 3 types of uncertainty when we try to find these subgroups. If I tell you about this. Now, if you look at the ARDS phenotypes, there is etiology, there is biology, and there is physiology. And there may be interaction of all these 3 subphenotypes or endotypes with each other, right? So how do you make these groups discrete? Okay. So one is the challenges to make these discrete is the missingness of data. That means we do not know what to measure. There's a failure to measure what is needed to be measured, either because we do not know what to measure or we are unable to do so. Continuity, that means inability to determine a break point on the distribution. So you find that based on the elastance of an ARDS lung, PEEP is useful versus not useful. Okay, but it's a spectrum. You find a spectrum, but you do not know. It's a continuous spectrum, and you may not know the break point where higher PEEP, a titrated higher PEEP may be better than a lower PEEP. So you do not have a discrete line to sharply define, okay, this is where we use this therapy, and this is the subgroup we cannot use this therapy. So there is no sharp line. There's too much of continuity in the data. Then the multiplicity. That means the problem of interest may have multiple domains, as I spoke about in ARDS. Multiple domains, multiple dimensions. Having more dimensions may help initially better understand the complex problems, but the data may be hard to reconcile and shrink it to guide discrete decisions. That's what I spoke about. And the testing of individualized therapies. And when you are trying to do that, when you are trying to find more and more discrete subgroups, to test individualized therapy against these small discrete subgroups could lead to very prolonged recruitment times for studies, increased complexity and costs of carrying out clinical trials. Those are the challenges. The other challenge would be incorporating multimorbidity in clinical trial design. That means in a critically ill patient, nearly a third of patients present with multiple morbidities such as hypertension, diabetes, COPD, and now complicated with multi-organ failure or more than a single organ failure. And the outcomes of these patients do not only depend on the acute organ dysfunction, but on the comorbidities, and the pathways interact in in a complex phase between the acute illness and the chronic disease. And this would be difficult to design a trial based on this. Then the other challenge would be to obtain real-time data in a critically ill patient, because critically ill patients have life-threatening emergencies, shock, respiratory compromise, and the need for urgency in care imposes a major barrier to advancing precision treatment, particularly if the time to do these assays of markers, biomarkers, or metabolomics or genomics takes long period of time, then it is too late to differentiate the subgroup. So that would be a possibility. And the next major hurdle is the managing the scale of clinical and biological data, the bioinformatics. Research in precision medicine is faced with an enormous burden of information, and the obstacles are the 4 V's: the volume of data, the velocity, the speed at which the data changes in a short amount of time and how you can analyze it, the variety of the data, the multitude of categories of data sets that arise, and the veracity of the data, the quality and reliability of the data. Even obviously we will be needing to use AI and machine learning, etc. But AI can produce good information only, garbage in, garbage out. If the veracity and the reliability of the data is poor, then what is coming out at the other end would be terrible. So that's why these are the challenges. And so these are what we have to look at when we are trying to define the subphenotypes and endotypes and response to treatment, the heterogeneity of treatment effect. Just look at the scale of data, right? So just for an example, a single cell sequencing in an individual patient, you are looking at the genomic data, the G, okay, G here, okay, then transcriptomic data, the proteomic data, the metabolomics of the microbiome data. Then you sample in multiple organs in an individual patient, and then you have to sequence, if not millions, thousands of patients of data set, and then multiple time points per patient per data. So all that is a huge volume of data which requires, I know it's not going to be very easy. It's an enormous burden of information. Now the concerns, these are the challenges. These are not insurmountable challenges, but they are major challenges. Now the concerns are data security and data, along with data sharing. On one hand, precision medicine cannot happen without immense amount of data sharing between stakeholders and having an open source collaboration, a scientific open source collaboration, but also it requires data security, so that protocols for anonymizing data is there, creating research ethics and protocols, and generating data sharing agreements. The other major thing, the other major concern, and which is very emphasized in this article in the AJR-CCM, is ensure that precision medicine advances be applicable to a broad range of patients across different healthcare delivery systems, resource-rich, resource-limited, racial, ethnic, socioeconomic, and demographic groups, and social and environmental practices. It's very important that we do that, because if you remember, if we realize almost 80% of the world's population resides in resource limited countries, and even in resource rich countries in resource limited regions within resource rich countries. And patients in these countries account for 85% in sepsis. If you take the example of sepsis, 85% of the global sepsis burden is in these resource limited regions. So if we do studies which do not look into for these patients and, or do precision medicine only for the rich countries and resource rich countries, then it is of no value, I think. And a threat to this goal is to solely focus on molecular substratification. If we base precision medicine on only the omics data, it is going to be very difficult, except in highly resource quaternary medical centers. So to do it in centers, we'll look at how we can overcome those concerns. So critical care outcomes vary significantly by race, ethnicity, and sex, and also the difference in lifestyle factors like nutrition, physical activity, exposure to drugs, alcohol, tobacco, etc. So how do we overcome these? The potential strategies to enact an equitable and just global access to precision medicine include: one is to consciously include diverse stakeholders from different geographic, economic, cultural, racial, ethnic, and gender backgrounds, or not only in the research committees, but in the advisory panels and the peer review committees also. Enhance efforts to promote the inclusion of ethnically, racially, and socially diverse population in studies and trials will allow broader applicability of precision medicine. Using approaches tailored for low resource settings, which includes basically developing cost-effective and rapid diagnostic tests which can be stored and performed easily in any healthcare setting. Using surrogate models that are reliant on readily available clinical data instead of complex biological data. If the complexity of the biological data is too much to differentiate the subphenotypes, in the near future, it would be very difficult to implement in resource-limited settings. Using AI applications that leverage high-dimensional clinical data with relatively low local technology, low resource local technology. The fourth one is called the fast second winner model. What it means is that some research somewhere in a developed country has shown that this intervention or this biomarker can be useful in differentiating, a subendotype, etc. Then using that, giving up the patent for resource limited settings at lower cost to another company to develop it, etc., or a different organization to develop it, allowing that also is going to be important. These are all part of distributive justice in healthcare. To end, precision medicine defines an approach, and this is in the article in Critical Care about precision medicine 2017. And I quote, precision medicine defines an approach in which the clinician comes to the bedside seeking to interrogate and understand their patient's unique physiology. While the terminology may be new, this sensibility harkens back to the earliest incarnations of critical care and remains at its core today. Not only critical care, but in medicine itself. We always try to look at the specific patient while we employ our ideas through our theory, through our guidelines, through our understanding of pathophysiology, to that specific patient as intensivist. This takes it to the next level. Precision medicine takes it to the next level. And it's also important to remember that medicine, as Rudolf Virchow said, is as much a social science as it is a biological science. The task of medicine is therefore not to merely to treat disease, but also to contribute to the health of the entire population. To make it, precision medicine should be for the benefit of all, not to the advantage of only a select few. Thank you so much.
Thanks, sir, for the insight into precision medicine. So,
I hope it was not too complex.
It is the future, sir, and you have shown us some light into the future. Can you stop the share screen? Will it be possible?
Yes, of course.
So, dear audience, if there are any questions, please put up the questions. The last point I think is very valid, sir, that most of this data is going to be generated in the West. And, a lot of the response to in sepsis in ARDS is genetically determined. And our genes, our means in the Asian population or the African population is definitely different from the Western genes. And whether that data will be applicable to the Asian population or the African population, I think is a cause of concern. And you have shown that.
So exactly. So what we have to do is, resource limited countries can share data, help each other, and already actually it's a possibility in terms of trying to find out our own genomic or even metabolomic profiles. Even if the genomics is difficult sometimes, it requires next generation sequencing. But there are many centers which can help you. Right now, the ICMR project which that we have applied for, we are cooperating between not only multiple centers, but we are cooperating with the Delhi Pharmaceutical and Research University, which has the equipment to help us analyze these. So what we are doing, like the ARDS trial I was still talking about. So what we are looking at, we are looking at therapy and doing the subgroup analysis by going back to the latent class analysis of Caroline Calki, looking at the markers which differentiate the two subgroups, and to see that that was a latent class analysis. Now we are looking prospectively that does it really differentiate the mark? These markers really differentiate the two subgroups, one, and do they respond to therapy differently prospectively? So these are all possibilities which we can do in this country. It is not, so yes, obviously the advantage will always be with the developed countries, but there are many things which we can do, and sharing of knowledge, sharing of database, and COVID to a certain extent has shown us that these are possible. It is how we look at healthcare in the world, how we put pressure on resource-rich countries to work together with us to find out these interventions which would be useful for different subgroups in different countries for different things.
Right. So then, sir, talking about something which is actually going on, this was looking into the future use of the different omics, which will take time. It's not going to happen so soon. Precision medicine in terms of, something like the management of septic shock or ARDS, which is also precision medicine. So, there are these guidelines, sepsis guidelines, which say
Right.
30 ml per kg, right? So these
Right.
are protocols, but looking beyond those protocols, everybody says now we have to use precision medicine. But for the ICUs as such, and especially for the junior intensivists, you cannot have precision medicine. Precision medicine requires a lot of understanding, requires depth, requires experience. So I guess
Right.
one has to, kind of have protocols only for the ICU, for the staff as such, everybody, and the senior intensivists can only use precision medicine. It's easy to say that precision medicine for everybody, but I think.
So yes and no, it's easier. But so when making, suppose for larger disease processes which are like sepsis and ARDS in critical care, right? So we can make within the groups, we can see that okay, septic shock, like requiring early ways of, I mean, the shock which is initial response to fluids, the shock does not, improves and worsens again after the initial response to fluid. Requiring high vasopressor, I mean, requiring vasopressors early would be the ones which would, so you can devise certain algorithms that, okay, would be ones which would do worse with higher fluids, use vasopressors more, right? And in that subset, I mean, these will take time to deliver. That's what we are doing at the bedside now as senior clinicians. We are able to teach our resident doctors this while we are, the whole idea of the fluid challenge, moving from a set amount of fluid to the fluid challenge technique earlier, fluid. So those are already, we are trying to see how is the patient responding to fluid. We are already doing some amount of it. So to protocolize it would be difficult, but not impossible. The patient not responding very well to fluids, but in shock, would require higher vasopressors, early vasopressors, probably would be the subset requiring vasopressin early. More analysis has shown that starting vasopressin early and reducing it late, outcomes could be better in that subset of patients. So all that I know it's not easy, but we could probably make algorithms in the near future, which could make it useful for a young resident doctor to do this. Obviously, this cannot happen in a completely resource limited setting. There would be difference in how a resource rich institute in a resource limited country like India will be able to manage versus vis-a-vis a resource limited setting in a resource limited country. I agree with you to a certain extent, but there are possibilities that can be done. Forget about the biomarkers, forget about those which are much more, expensive, etc.
Yes, sir, I agree. If one tries, one can probably make some kind of a protocol with precision medicine also, but needs a lot of hard work. Okay, sir.
Yes, sir.
I think that's all then. Thank you so much, sir. Till we meet.
Thank you.
Thank you so much.